Okay, this is lecture 4 in the cell pathology and infection module, but the cell pathology lecture specifically. This is part 3 of that. We're covering the genetic changes leading to cancer, we're covering the hallmarks of cancer, and in the previous 2 lectures we've covered EGFR and cell sufficiency of growth signaling and PRB, and we've just covered telomerase, and we've covered evading apoptosis. What we're now going to cover is the VEGF axis for sustained angiogenesis and invasion and metastasis. Okay, so for tumours to grow beyond a certain size, they must have their own direct blood supply. And the majority of cancers that you will study are epithelial cancers. And epithelial cancers by definition, or epithelial cells by definition, do not have their own direct blood supply because the blood supply is in the connective tissue or mesenchyme, and the epithelium is hidden behind a basement membrane. So for a tumour cell to be able to grow beyond a certain size, it has to have its own blood vessels in close proximity to the epithelium. And what we can see on this diagram, this is a small ductal carcinoma in situ, so it's a pre-malignant stage of breast cancer. And what we can see is it's a smallish lesion, it's a duct that's packed full of tumour cells. It's still got an intact basement membrane, but around it are blood vessels, stained brown in this case, and those blood vessels are delivering oxygen and nutrients to that small developing tumour. But this can't get any bigger. The only way this can get any bigger is if those tumours break out of that basement membrane to access the blood supply and then be able to contact those blood vessels directly. So a tumour can't get any bigger than about 1 millimetre cubed without its own integrated blood vessels within that tumour. So this is another bottleneck to tumour growth. Now the reason the blood vessels are around this particular lesion is because the tumours themselves are producing vascular endothelial growth factor, and that is a growth factor for vascular endothelial cells. Blood vessels are lined with endothelial cells. So if the centre of this ductal carcinoma in situ is hypoxic, that is low levels of oxygen, that is a signal for cells to produce vascular endothelial growth factor, because if a cell is hypoxic, it needs more blood vessels around it. So it will release VEGF, that will encourage blood vessels to migrate into that area and feed that bit of hypoxic tissue. The tumours become very readily hypoxic and therefore produce vast amounts of VEGF, and therefore they can manipulate the vasculature to feed the tumour. So this slide summarises the pro-angiogenic signals that tumour cells produce in response to low levels of oxygen, also known as hypoxia, and this can particularly include vascular endothelial growth factor, which recruits the endothelial cells, but also fibroblast growth factor and platelet-derived growth factor, so that recruits in the pericytes and fibroblasts that will stabilise these vessels. So VEGF is another tyrosine kinase receptor, very similar to other growth factor receptors in that way, and the VEGF protein will bind to the VEGF receptor, and that will activate the endothelial cells to proliferate and migrate towards the source of that VEGF. So the tumours themselves remain generally hypoxic, and that drives the angiogenic process. So the final hallmark is invasion, which subsequently leads to metastasis. So what we're dealing with here is initially a primary tumour. A primary tumour is the tumour at the site on which it initially forms, and then a metastatic tumour is at a distant site, typically via lymphatic or blood-borne dissemination from the primary site. And the primary tumours account for about 10% of death, 90% die from the metastasis. And what we can see is that the larger the tumour is, the more likely it is to have already metastasized. So very small tumours are far less likely to have metastasized. A very large tumour is very much more likely to have metastasized. So this is why finding tumours earlier is much better in terms of a prognosis, and this is related to tumour stage that we've talked about. A big tumour, 10 centimetres, will be a T4, whereas this was likely to be a T1. So this is why survival is so much better for small tumours than larger tumours. So many primary tumours are resectable and curable by the surgeon. However, metastatic tumours that have left the primary site are much more difficult to treat because they are rarely able to be cured by surgery, only in a very odd exception. And that is why you have to then have a systemic drug approach rather than a surgical approach. So this is just showing disseminated non-Hodgkin's lymphoma, and everything in yellow on this scan is tumour material. So for a tumour to be able to break away from the initial tumour site to a distant site, it needs to undergo or overcome the metastatic cascade. So at point 1, you've got a primary tumour initiating angiogenesis. It does that by switching on vascular endothelial growth factor. That encourages tumours to be well vascularised, and the tumours themselves have had to break down the basement membrane to allow the tumour cells out or the blood vessels in to feed that tumour. Then you've got localised invasion, where the tumour cells have to invade through the surrounding connective tissue, and then intravasation, which is entry into a blood vessel. Now, blood is a very alien environment to most tumour cells, certainly to solid tumour cells, and lots of tumour cells that enter blood seem to undergo rapid apoptosis. And we're not entirely sure why, but they do. It's a very hostile environment. So it's a very rare and special cell that can survive this process. Then the tumour cells migrate around the body and arrest at a distant site. And this is often a distant site that has a very dense vessel bed, large blood flow going through it. So lungs, bone marrow and brain are very frequent sites of metastasis, but then so is the liver as well, which also is similar in terms of blood flow. So some tumours seem to like certain distant sites. So colon cancers predominantly metastasise to the liver, and that's partly due to the hepatic portal system that delivers them straight there. Ocular melanomas, rather obscure tumour, they always metastasise, if anywhere, to the liver. Prostate cancers tend to go to the bone marrow and lungs. So do breast cancers. They often go to bone marrow or brain. So different tumour types have different favoured sites of distant metastasis. A lymph node is a very frequent place for them to go. Once they get to that distant site, something encourages them to extravasate, that is, creep out of a blood vessel. Sometimes it's a growth factor that is produced at that site. So we think that there is a chemokine called SDF1 in the bone marrow that encourages prostate cells to extravasate out of the blood vessels in the bone marrow. The tumour cells seem to express a receptor for that chemokine, and that encourages them to creep out of the vessel, specifically in the bone marrow. And that's partly why prostate cancers can form bone metastasis, so that's the hypothesis at least. Once they get to that site, let's say the bone marrow, they can then form a micrometastasis, and then that micrometastasis has got to initiate its own blood vessels to then form a secondary tumour at that distant site. So that's a lot of steps that a tumour cell has to do, and a lot of new functions that it didn't previously have. So it is a very rare cell that metastasizes. So just to illustrate that, here is an epithelial tissue. These are epithelial cells. This is connective tissue. This is where the blood vessels are. This is where the epithelium is. And if these epithelial cells are going to access the blood supply, they've got to invade through this connective tissue. And then if these cells become hyperproliferative but without the ability to invade, they will fill up this duct, but they will not get any larger, because cells have to be within 0.5 millimetre of a blood vessel, and if they cannot get their own blood vessels into that tumour, it just can't get any bigger. So early tumours are limited in size by the inability to access their own blood supply. So this is illustrated here. These are more carcinoma in situ with intact, so this is breast ductal carcinoma in situ with intact basement membranes. So you've got bits of connective tissue in between, and that's different to an invasive carcinoma. So this is non-invasive, premalignant. This is invasive, malignant. So degradation of that basement membrane is one of those rate limiting steps, and that basement membrane can be rich in collagens and other cellular matrix proteins. And what we know is that enzymes such as matrix metalloproteinases, MMPs, 2 and 9 in particular, they are very good at breaking down this basement membrane. Other secreted enzymes are also able to do this. So you can have a cell which starts to express matrix metalloproteinases that can break down that basement membrane, allowing those cells to creep out, and you end up with a disease such as this. But breaking down that basement membrane is not the end of the story, because cells are effectively stuck to each other. The cells are very sticky, or most, certainly epithelial cells and connective tissue cells are very sticky. Immune cells can obviously slide in and out. And the reason why is because those cells have cell-cell anchoring junctions and cell extracellular matrix adhesions. Specifically, the cell extracellular matrix junctions are integrins, and if cells change their integrin expression, then can be associated with a more invasive phenotype. So, for example, we know that alpha-2 beta-1 integrin is associated with metastasis in many epithelial cell types, and that's because they would normally be expressing other integrins, and as they change to alpha-2 beta-1, that allows them to slip away from their normal extracellular matrix links. So that's the cell binding to the basement membrane or other extracellular matrix, which is going to keep that cell in its place. But equally, the cell next door is going to keep it in its place, because cells express cadherins and components of these desmosomes. All of these cell-cell adhesion complexes keep cells in their right place. So the only way that a cell can slip away is by changing these adhesion molecules or adhesion proteins. So this is shown here. This is a normal melanocyte. Melanocytes interact with keratinocytes in skin, and they express E-cadherin. So normal melanocyte in skin is expressing E-cadherin. Upon transformation, so conversion to a melanoma, E-cadherin is lost and N-cadherin is then expressed. And by expressing N-cadherin, that allows that allows the now melanoma cell to interact better with fibroblasts and endothelial cells and no longer be tightly bound to the keratinocyte. So this allows the transformed melanocytes to slip away from their keratinocytes, which they're normally bound to, and then start invading deeper down into tissues, and that would be a key change that occurs in invasive melanoma. Now, in the first two parts of this lecture, I've talked about mutations of EGFR and mutations of P53 and PRB. So they were very much definite genetic mutations. Many of the features associated with angiogenesis and adhesion and invasion tend to be changes to gene expression rather than specific oncogenic mutations to the key genes. So you don't necessarily have mutations to E-cadherin, the cancer cells just stop expressing E-cadherin. And this final slide illustrates effectively a multi-hit model of how cancers go from being normal to cancerous, and the genes that they may have either mutated, activated, inactivated, or overexpressed to achieve all of the hallmarks of cancer. So it has got each of the components and an example mechanism, and the order of events varies between tumours. Sometimes, like in colon cancer, it's a very predetermined order of events. Sometimes it's a little bit more random. So you could have a cell where the first thing is activation of RAS, the second thing is loss of retinoblastoma, the third thing could be a change to tissue invasion and metastasis, so inactivate E-cadherin. Then you might get a bit of angiogenesis formed by inducing VEGF expression in an environmental response to low oxygen. Then maybe you've got evading apoptosis. I've not talked about pro-survival factors, but it could be something that causes survival rather than prevents death, and so on. So each cancer can achieve all of these hallmarks in a slightly different order. Sometimes it's fairly predictable, sometimes it's completely unpredictable, but what we do know is that all of those hallmarks of cancers, at least for solid tumours, have to be overcome before a tumour, a cell can develop into a transformed cell, a premalignant cell, and then a fully malignant cell, and then an invasive and metastatic cell. And this takes the concept a bit further with the 10 hallmarks of cancer. So initially the 6 hallmarks of cancer were published, and then 4 more were added, including genome instability, which is the mutator hypothesis, deregulating energetics, so this is looking at glycolytic shift, looking at oxidative phosphorylation versus glycolysis. There's immune destruction, and there's tumour inflammation. And in all of these hallmarks of cancers, what we have now got is either drugs or drugs in development that specifically target the defect that is achieving one of those hallmarks of cancer. And if you can prevent one of those hallmarks of cancer, then there's every chance of stopping that tumour from either progressing or growing, and hopefully encourage it to regress. Now, what we're going to do throughout the rest of this module is specifically cover things like EGFR inhibitors as an anti-cancer strategy, so we'll cover that in later lectures, and we will touch on some of these others as we go throughout the module. Okay, that's it for the end of this session. So that's completion of week 16.